welcome to today's STERIS Tech Talk on executing a successful accelerated and real-time aging study. STERIS Tech Talks are a series of webinars covering subjects relating to gas and radiation sterilization processing and the laboratory testing and validation services which support these processes. My name is Ashley Marute, and I'm the Associate Product Manager for STERIS Applied Sterilization Technologies. I'll be the host for today's event. Our presenter today is Noel Gibbons. Noel is the Technical Advisor for Medical Device and Pharmaceutical Packaging within STERIS AST. He has significant experience in a wide variety of areas from manufacturing, process validation, package testing, packaging process validation, package integrity testing, quality, and international test standards. He has significant practical experience in medical device packaging at all levels, including evaluation of proposed packaging and current packaging designs and related validation documentation. Noel has presented nationally and internationally on medical device and pharmaceutical packaging testing. All attendees are on mute for the presentation. However, we would like to encourage everyone to submit questions using the questions function on the GoToWebinar control panel. Questions will be answered following the presentation. Today's presentation will be recorded and uploaded to our STERIS Applied Sterilization Technologies YouTube channel. Please note that continuing education credits are not provided as part of this webinar. And now over to Noel to begin the presentation.
Thanks, Ashley, and welcome to this Tech Talk on effective performance of real-time and accelerated aging studies. Basically, when you start to look at real-time aging and accelerated aging of a product, you need to understand the actual product life cycle before you can actually make a good decision on how to age your product and how to actually carry out a shelf life study. You need to understand basically the concept from design, which includes ergonomics. And by ergonomics, I mean how the product actually fits into the actual packaging. Is it a tight fit? Is it a loose fit? Is it going to be moving around inside the actual shipper box during, say, the likes of distribution? Can it cause any issues with the seals, which could deteriorate the sterile barrier? From design to prototype and onto manufacture into the sterilization processes and validation of those processes, onto the shelf life and storage conditions. And then also from your sale to initial distribution into sub-distribution and out to the end user, and what the impacts that will be on the actual packaging or the product. You also need to look at the questions, and you have to answer these questions to define a proper and a good shelf life study. What is aging? What is the purpose of aging testing? What causes packaging and product to age? Do I need product in the packaging for the aging studies? What is the glass transition and melt temperatures of materials? What about relative humidity? And when do accelerated aging and real-time aging have to be carried out? What risks are involved? And how long is the shelf life of your product? We're going to try and answer all of these questions throughout the presentation. They won't necessarily be in this order, but please bear with me. Other considerations you need to take into account is where will your product be stored before being used? Is it going to be in your own warehouse? Is it in a distributor's warehouse? For how long is it going to be stored for, and in what conditions? These are all prime areas that you need to know and understand. You also need to know and understand your packaging as well. Is it going to be capable of being aged? For example, the picture here on this slide shows a product that traveled just 80 kilometers in the back of a courier's van. As you can see, it didn't last very long in that short period. So how is it going to react over a long period of time of 1, 2, 3, 4, or 5 years? Will it perform reliably for its expected lifetime is a big question. And my answer looking at that picture would be absolutely no. In relation to shelf life risks, every process has risks. When you get out of the bed in the morning, until you go to bed again then the following night, or whenever you go back to bed, there are risks. Some of them can be significant. If you look at, say, the likes of a manufacturing process around packaging, the manufacturing process has risks in how the product is manufactured, how the sterile barriers are created, the packaging materials itself, how that's handled. During the manufacturing process or the packing process out into distribution, whether altitude has an effect on it, and into the global distribution locations, which will pretty much determine your ambient temperatures that would you normally use either for storage or for your calculations in accelerated and real-time aging. Also your storage and your shelf life, how, where and when. These must be assessed to determine when and why failures may occur on your package or your sterile barrier. And you should never underestimate the packaging challenges that can occur during a project, because they will always come back to bite you if you don't have them assessed. If you look at, say, ISO 11607, Clause 6 states that the packaging system shall provide physical protection and maintain integrity of a sterile barrier system and maintain this sterility to the point of use or until the expiry date. But when we mention the point of use, that's not when it leaves your back door as your manufacturing site. That's when the product gets to the bedside of the patient and is finally opened and the product removed. It must retain its sterility until that time. And it must maintain its integrity, the packaging integrity, over that entire duration. Accelerated aging protocols are regarded as sufficient evidence by the regulatory bodies for claiming expiry date, but that's only until the real-time data from real-time aging studies becomes available, which can be 1, 2, 3, 4, or 5 or 10 years down the road. If you look at, say, ISO 11607, again, I have a graphic here of pretty much the whole process for packaging. What you have here is everything from your DQ, your design qualification, IQ, OQ, PQ processes and what's involved in them, into your production processes, your sterilization processes, again, the likes of usability studies and shelf life validation, which covers both your accelerated and real-time aging. And again, how you prove that the package integrity has been retained over that period of time, either in real-time or accelerated aging. What is shelf life aging? Shelf life is the length of time that a product may be stored without becoming unfit for use, unfit for consumption, or unfit for sale, and demonstrates the ability of the packaging system to maintain a sterile integrity and steel strength over that defined period of time. This is normally indicated by a label on the product, which defines the actual date that it has to be used by. It's normally on the primary packaging, but maybe on the secondary and tertiary packaging as well. And it's a lot handier for use and for auditors if they're looking at that. Shelf is determined not by me in the test lab, not by the regulatory bodies, but by the manufacturer of the product. And it's not solely on the chemical makeup of the materials. However, most of the accelerated aging theory is based around the chemical reaction and the Arrhenius reaction rate, which we'll go into in some detail later on. These processes include, as I mentioned, the manufacturing process, the product material types, the type of packaging it's contained in, how the product is to be stored, the packaging product dimensions, and the sales and marketing team. And the sales and marketing, or the marketing team more so, will have a very big input into the shelf life of the product, because they're the people who are going to define basically how long this product is going to be on the market. So you have to blame them for a 5 or a 10-year shelf life, which can be good in some ways. They will define what that is, and they need to take that into account as well. What is aging? If you look at the definition for accelerated aging, it's an artificial procedure for establishing the shelf life to estimate the useful lifespan of a product when the actual lifespan data is unavailable in an expedited manner. Basically what that's saying is that you can use accelerated aging until your real-time aging data is available. It's conducted by simulating a simulated exposure of the product to elevated temperatures and humidities where necessary, for the equivalent period of time to be claimed as for the product expiration date. And as I said, it's only tentative until the real-time aging data becomes available. I'll be repeating this a number of times during the presentation because it's a very important factor that you have to take into consideration. In relation to real-time aging, this is the stability testing process that provides real data to determine the shelf life and the effects of aging on materials, and is normally done at ambient temperatures and relative humidities. Typical ambient conditions that would be used for real-time aging will be 23 degrees centigrade with a 50% relative humidity. That level of temperature can change depending on where you are. Typically, the ICH has done a study a good number of years ago and broken up the ambient areas for around the world. So you can have anything from 20 to 30 degrees as your ambient temperature, depending on the country you're in or the zone you're in within that particular country. New or modified medical device products must be storable for their defined shelf life and to the point of use without deterioration. So what's the difference between real-time and accelerated aging? The main difference is that the temperature levels and humidity levels that are actually being used. For accelerated aging, the temperatures are elevated and the relative humidity levels can be elevated as well, but you need to be very wary when you're doing this. However, the accelerated aging is only a guesstimate of what's happening in real life aging or real-time aging. And sometimes, very rare that you get it, you will get accelerated aging to pass all its tests, and you'll be able to prove that it's retained its integrity or sterile barrier, and you may get failures after real time. And it can be very difficult to answer the questions around that. Whereas the likes of real-time aging is carried out, as I said, in typical ambient or nominal storage conditions with normal temperatures, as I said, in around 23 degrees with 50% humidity. But they do demonstrate how the packaging sterile products reacts over time, and that's what's taken into account by the regulatory bodies. Artificial aging is the process to simulate this and speed up the normal aging processes to release product to the market in a short duration. So basically, if you have a product that's going to remain on the market for 5 to 10 years, you can't wait for 5 years to release that product because it just wouldn't make sense. So you want to be able to release it as soon as possible. And we'll have a look at some of the numbers and calculations around that shortly. There are also other accelerators that cause aging, such as the likes of sunlight, oxygen, heat and temperature, which we're going to concentrate on, vibration, chemical reactions, and relative humidity levels. As I said, accelerated aging is optional, but real time is required when establishing an expiration date. And again, for the very simple reason that I've already mentioned. Regulatory bodies will only recognize the accelerated data as acceptable until the tests can be repeated on the real-time age products, where the primary focus is on the sterility being maintained to the point of use or until the expiry date. And where the challenge is for the additional testing that is performed after aging to assess the packaging and the product. These will be typical like visual inspection, dye penetration testing, bubble leak testing. Again, we'll have a look at those towards the end of the actual presentation. Accelerated aging calculations are based on the Arrhenius reaction rate, or Arrhenius equation, as per the ASTM F1980 standard. And this simply states that any increase or decrease in temperature will cause a double or halving of the temperature, or the reaction rate, not the temperature. So if you increase the temperature by 10 degrees in temperature, it'll double the rate of the chemical reaction. And the premise for the deterioration of the material, as I said, is due to a chemical reaction. And the 4 variables that are involved in this accelerated aging calculation are the elevated temperature or test temperature, the ambient temperature, the Q10 or reaction rate factor, which we'll look at again in a minute, and the real-time equivalent days or duration. in years, and they're all plugged into the formula on the right hand side there. Typically, the Q10 value is set to 2 as a conservative level for medical devices, and it's what the majority of companies use. If you want to change or move outside that, you're going to have to justify it, and you have to justify it by showing that the product and the packaging has not been degraded and in significantly greater detail than it would be in normal aging. And the level and nature of the damage with this must be similar to that reported in literature to ensure the reaction of the coefficient and accelerated aging temperatures are maintained. Again, the assigning of an appropriate Q10 factor is the responsibility of the manufacturer, not to me, not the lab. You can take a Q10 value of 1.8, 2.2, up to 2.5, or any of the any number in between. But you must justify that. I cannot do that from a test lab point of view. The accelerated aging factor is defined as the ratio of real world lifetime to test duration. You're using the Arrhenius reaction rate. So first you calculate your accelerated aging factor. And basically what that's done is 2 to the power of your accelerated aging temperature minus your real-time temperature divided by 10. And that's then plugged into the accelerated aging formula and divided basically into the real time that you want to actually assess. This increase in the rate of chemical reaction that has caused the accelerated aging, and it shall be performed prior to any distribution of the product. Because if you haven't proven that your product can or does have a shelf life over a short or long period of time before you actually start to release it to the market, you're on a loser straight away. This is a chart I did up a number of years ago to depict the different temperatures versus aging time in weeks. So basically, what you have is you have on the left-hand side you have the number of weeks, or time in weeks, and down the bottom you have your accelerated aging in degrees centigrade. So if you take typically a 22 or 23 degrees ambient temperature, a year will take approximately 52 weeks. Whereas if you increase that temperature, say with an ambient temperature of 23 degrees, that 1 year will be reduced to the equivalent of 4 weeks at 60 degrees. If you change the ambient temperature in the calculation again to 22 degrees, you have at the 60 degree level, you're down to 3.7 weeks. So again, it's a saving in the time in the project. However, that brings additional risks into place as well, which we need to be able to take into account. So typically we're looking at an accelerated aging calculation. From the actual formula, you can manipulate what you're actually doing. Say if we take an example, an elevated temperature of 55 or 50 degrees with a 25 degree ambient and a Q10 value of 2. 365 days can be done with an accelerated aging in 65 days. So it's a huge saving in the introduction of your product. However, if we manipulate that formula and just change the ambient rate by 2 degrees, again with all the other figures staying the same, we're now down to 57 days. Again, another saving in the actual whole study. If we increase the accelerated aging temperature to 55 degrees with an ambient of 25, all of a sudden we're down to 46 days. And that keeps going on and on and on, the more you play with the figures. Up as far as 60 degrees, which is the highest recommended for within the standard, because the calculation becomes non-linear after that. So if you look at there, the last one there is 60 degrees with a 23 degree ambient, you are achieving a full year in 29 days. The accelerated aging and the real-time aging must or should be carried out start within 3 months of each other. So as you're actually achieving a relative alignment with the product and the packaging that's in use at that time. So you're using the same lots, the same batches of product, the same batches of packaging. However, what you must take into account is saving this couple of months shown in the tables above. Is that worth the risk? And you have to define what that risk may be. If you look at, say, the two pictures here in relation to shipper boxes, the shippers started to fall apart in the aging chamber after a relatively short period of time. For two reasons: that the temperature was very high and the relative humidity within the actual shipper box, or within the chamber, was very, very high. And what that did is actually destroyed the actual adhesive in the manufacturer's joint, and the box started to fall apart. If this was a real-time scenario where your product was actually being shipped out in the field and exposed to very high humidity, this could also happen. But what will happen in that case is that your courier will take the product and they will deliver it to its end point, but it'll be wrapped in their lovely packing tape, which doesn't look good for you or your product. The question you have to ask there: how high is too high, and when will the material start to deteriorate in the actual packaging or in the product? Like if you have, say, the likes of a drug-eluting stent or something like that, your accelerated aging temperature can only go, say, in the region of 35 to 45 degrees. You must balance the project time needs over the risk of test failure, because if you're not doing that, then you run the risk of failures in your aging, and you have to start again or justify what you're doing. In relation to the materials, what is the melt and the glass transitioning point of materials? The melting point or temperature, or TM, of material is a critical point above which the crystalline region in a semi-crystalline polymer are able to flow. So basically what that's saying is that it goes from a solid basically to a liquid. And once it goes to that state, it will not recover. It will not go back to its original state. Okay, the TM for crystalline, semi-crystalline polymer is much, much higher than the glass transitioning point, or TG, for a particular product. We'll have a little bit more look into that shortly. The glass transitioning point on a polymer is the transition point from a soft rubbery state into a rigid or glassy state. So when you're forming your actual tray, or forming your pouch, or forming your actual product, it's when the temperature that the material becomes solid, and vice versa. If you're overheated, it will actually start to melt again. If you look at the actual picture there, what's there is actually a cutout of a product cover that was on a customer's product. They put it in at a specific temperature. It was a lovely straight piece of material when it was in, depicted by the red line, but after a couple of weeks in the chamber, it was nice and curvy. Which was no good to the customer, so they actually had to start again. So they're the actual risks that are there. So they'd lost the product, they'd lost the time, they'd lost the actual dates they had for their submission to the regulatory body for this particular product. And again, you have to load the specification of the material, and in this instance, it wasn't actually assessed. If we look at, say, the glass transitioning point here on the right-hand side, or the left-hand side on the graph, you have your glassy state, which is your solid state, and in the middle in there between the glassy state and the rubbery state is your glass transitioning point. Once you go past this, you're actually starting to damage the material to a point where it's not going to recover it to its original shape or statistics. If you go down to the TM and below TM, you're actually going into a viscous state, so you're actually melted the material. And again, it's not going to recover to its original state. If you look at, say, typically the PET or PETG there on the bottom of the table on the right, which is the lowest temperature, typical temperature that for a material, you're looking there at between 73 and 88 degrees for this product. And if you're getting up around that temperature with your aging, you're actually going to destroy the package or the product, whatever this material is actually incorporated into. If you look at the materials here in the pictures, as you can see, the customer or the designer of this didn't take into account either the glass transitioning point or the melt point of the actual package and or the product. And it took longer to actually clean out the chamber after the test than it did to actually do this damage. And needless to say that the aging failed. Again, because the glass transitioning point of the polymer had been exceeded drastically. In relation to relative humidity, while it's not a factor in determining the aging duration, especially in the calculation for in the Arrhenius reaction rate, it is a real life factor, and that has to be taken into account in relation to, say, the likes of polymer degradation, so like the likes of hydrolysis or corrosion in metal. It should absolutely be in there. Or like the example I gave earlier, the boxes, the shipper boxes actually falling apart. But the problem is you need to have a realistic level of humidity in there. And what that is can be difficult at times to decide. Typically, if you're going to have a very high humidity level, you're going to create artificial defects that are not time related. So, for example, the box falling apart would be one of those, or the box softening, and the whole package collapsing when there's a load put on it. If you go the opposite way, where you're putting a low level of humidity in the box, which some customers do, they try and go down between 5 and 11%, which you can do when you extrapolate the formulas or the graphs in ASTM F1980 down. But the problem here is that there are very few places on Earth that you're going to get a 5% relative humidity. And there's no point in doing that, because it's not going to give you anything that is time related to a time related stress, I should say. Typical levels that are chosen should be chosen to avoid any significant damage to the packaging or the product. And typical levels that are being suggested will be between 45 and 55 degrees or percent humidity. Again, what you're trying to do is avoid damaging your product unnecessarily. What I put together in the next slide is a typical guidance or summary of how the whole aging process should be carried out, the way I see it being carried out. Basically, the simplest way is to select your Q10 value. Typically, as I said, the conservative value here will be 2. Define the desired shelf life, or the real-time aging or age of the product. Define the aging test intervals, and including the time point 0 there, so your starting point. So you assess your product and packaging at that point. Define the test conditions, say for real-time aging. Is it going to be 23? Is it going to be 30? Is it going to be 20 degrees? And what humidity levels you're going to include in there. Again, it needs to be realistic in that. Define your accelerated aging conditions, what they're going to be, and what temperatures that's going to be. Is it 50? Is it 55? Is it 35? Whatever the case may be. And decide if humidity conditions are going to be required and what levels. Then calculate your test duration for your accelerated aging, and define the sterile barrier system properties. What you're going to prove is good after the accelerated aging and real-time aging has been completed, and how you're going to prove that it's a success or not. You're going to age then the samples in your aging conditions, your accelerated aging conditions, and the same thing in your real-time conditions. Then you're going to define your acceptance criteria, how you're going to say that it's passed or failed, and evaluate the barrier after the sterile barrier after both accelerated and real-time aging. There's no point in just doing, say, the likes of a peel test or a bubble leak test and not comparing it to anything. So what I suggest there then is that you compare your results back to both the T0 results and the PQ results from where you've created your seal in the first place, to see if there's any significant deviation or deterioration in the seal strength over that period of time while it's been in its aging, or during the real-time shelf life. If you look at, say, the likes of the post-evaluation testing, what can you do? The typical one that's done by most companies will be the likes of the visual inspection, typically to ASTM F1886. Basically, what you can do here is determine channels, leaks, breaks, cracks, melted product, melted packaging product that's come out of its place. It's a quantitative measure which gives you an accept reject rate of the unopened packaging. It gives you an attribute that can be linked directly to variables in your process, such as the seal parameters, the equipment that's been used, the material that's been in the product and the packaging, as well as the environmental conditions that it's been exposed to, whether it's been underexposed or overexposed. Typically, these visual inspections will show you any product that's caught in the seal, seal creep, where you're getting an overheated product, or where you're having, say, the likes of an effect of the product inside affecting the packaging itself. Again, you're going to see these whether it be in the likes of accelerated aging or real-time aging or distribution or any other processes that you're actually putting through. If you look at the likes of the integrity or seal strength tests, what you're going to do the testing to will be the likes of the ASTM F88 standard, or alternatively the EN 868 Part 5 test standard. What you're trying to prove here, or to show here, is that you've maintained a minimum seal strength and that you have consistency, including repeatability and reproducibility of the actual seals after your aging processes. So at the base, after 5 years sitting on a shelf, is it the same as when it was actually manufactured, or is it slightly below what it is? Or sometimes the materials can harden and actually go slightly above what you originally got. And again, also to document the results. Typically, what you're looking for in a seal strength validation at this point is that you're someplace in or around what you defined as your nominal seal strength, or wherever you'd actually started the aging study. If you had gone down, say, 5 or 10 degrees below the nominal seal strength to create your seals, then that's where the seal strength should be at the level it should be, or above, again, depending on which worst case you've chosen. But it shouldn't go below the lower spec limit or the upper spec limit for that seal. The likes of dye penetration testing, an excellent test which will show you very, very quickly what is happening with your seals. Depending on which test you're using. It's the likes of the ASTM F1929 standard will look at all your seals. It'll give you an answer to see if you have any channels in the seal or any openings in the seal in less than a second. And you can do a complete test within 20 seconds, depending on the size of the actual product. The larger the product, the longer it's going to take. The problem with it is that the dye with the surfactants in it will cause both wicking and leaching of the dye solution out through some packaging, depending on the properties of the packaging. And you can lose sight of a defect that you found very, very quickly. In relation to F3039, it's normally used for the likes of foil and foil pouches, and there are 2 test methods in there, one for the seals and one for the surface, and both are deterministic type tests. Problem is that they're subjective, and the operator needs a lot of training to see and know what exactly they're looking at, and is it a failure or is it not a failure? And I'll show you why in a minute. But the advantage of this is that it's been validated down as far as 50 micron. So typically what you're seeing is here is in both pictures is the dye been introduced into the actual packaging. That's method A in ASTM F1929, where the dye is injected into the actual pouch, and you'll see that it's actually trying to leach out and wick out through the actual seal. In relation to the false positives that you can get from container closures or packaging like this, if you get a crease in the seal, it can look like it's an actual failure. So you need to go and investigate that in much more detail. So again, as I said, the operator and the technician that's doing the actual testing needs a lot more training than normally would be required. In relation to one of the last tests I'm going to talk about today is bubble leak testing. It's probably one of the best tests that I believe in. Again, it's a deterministic test, and it basically tests your whole package. And by whole package, I mean it tests the surfaces and the seals, not just the seals like the previously mentioned standards. The problem with it is that test sensitivity is only down as far as 250 micron, a defect of down of 250 micron. But again, that negated by the actual being a whole package integrity test. The problem with any testing like this is what you need to determine what is you're going to accept and what are you going to look for. You can go down the lines of doing the likes of helium gas sniffer type testing on packaging and to see if you have a defect down as far as in around 1 micron. But is that going to gain you anything? And again, that's all in the risk assessment that you have to define. What you have to remember is that your packaging is part of your product, and it's key to protecting the patient against any damage or deterioration in their condition. So anything that you can do to safeguard the integrity and sterility of the packaged product is the better for the inpatient.
Thanks again for joining us today. This concludes the webinar.